So we are recording. Microphone is on. I charged it last night. And the adapter. I'm charging them both together now. Alrighty. So this stuff, I don't know. I love it. And maybe I'm weird. I really enjoy the energetic conversation. I enjoy the physics behind it and all that fun stuff. So I've been told from many people that you either like the genetic stuff or you like the metabolism stuff. You usually don't like both. I don't understand that. I like both. So I'm going to try to make them both equally exciting for you. Um, but maybe I have a slight preference for this stuff. Now, I'm supposed to make you guys memorize like every single step of this reaction. I'm not gonna. We're just gonna leave that to ourselves because the one class where I feel like you really need this is microbial physiology. And he goes over all of this for you again. And in the end, what you have to really learn are the intermediates of the TCA cycle, which in the end aren't too hard to learn, especially like sitting with it for a while. So if you see it here, you've seen it in 106, 107, you're going to see it again if you take microbial phys. You'll get that stuff there. This is not going to be the last time you see it. And then in the end, if you need it for a lab, you can pull it up and look at it. So I'm not a big fan of making you guys memorize what's coming up. I'd rather you guys understand what's happening and realize that yes, we're taking a 6-carbon molecule, breaking it down to a 3-carbon molecule, and then slowly breaking apart those carbons even more to stepwise release the energy. That's the main takeaway here. I want you to understand the big picture and not get caught up spending, time until 3 a.m. memorizing crazy chemical names. So maybe that means we're going to end a little too early today, but we'll see. We'll see. This part is not going to be as in-depth as tomorrow's lecture. Tomorrow's lecture, we're getting into the electron transport chain and photosynthesis, and for that, we will kind of need to know a lot of the steps and the mechanisms, but I feel like that is better than memorizing chemical names anyway. So let's dive into it. There is a whole energy discussion. The book is going to go a lot deeper into it than we do, but just know that I want you to understand the big picture of it, and you can wheedle out the details as needed in your future classes, because in the end, if you know the big picture, you're good to go for everything else. So that's where we're going to stay, is more in that big picture area. Okay, why is this not working? Oh, that's why. Sorry, guys. To say that like life is crazy right now is an understatement. And it's my anniversary and my husband's birthday this weekend. Thank you. Yeah. So it's just like lots going on. All right. Now there we go. So lots of key terms, but I think you've seen a lot of these before. And a lot of them are pretty straightforward. So we talk about energy. Everything needs energy to live. Mainly because of entropy, which we're going to get into a bit today. The universe is constantly getting more and more chaotic, and as living things, we are incredibly ordered. So we have to build the order out of the chaos, and that's not exactly easy. It requires a lot of work. So there's 2 basic sets of energy requiring reactions in the body, or in the cell too. It can be catabolism or anabolism. Normally when I teach this, I'm teaching it in like anatomy and physiology terms. So I always forget, oh yeah, we're talking about cells here. So catabolism is the breaking down of molecules to get energy from them. This is eating. Anabolism is building new molecules. So we break down our food to get energy, to get building blocks, and then we take that energy and building blocks and make new DNA, make new proteins. That's anabolism. The breakdown is catabolism, and together we call it metabolism. And all of this energy ultimately comes from the sun. So all the energy that we have to do everything in our bodies comes from what we eat, and that either comes from what they eat or from the sun, because the plants get it from the sun. There are a couple asterisks on it that we're going to talk about with things that get their energy from minerals and inorganic sources, but for the most part, even that kind of comes back to the sun in one way or another, because all of the earth is effectively stardust anyway. So, all right. Any questions about this before we truly dive in? I'm going a little bit slow because I know as the semester goes on, we like to come in a little later because we're like, I can sleep in 5 more minutes. All right. Now, energy, the physics definition is the ability to do work. So let's take that and apply it to bio. What is work? It's fighting off infections. It's reproducing. It's moving. It's building up cells. It's doing the work of digestion. It's all of those things that we do to stay alive. And the other side of this is entropy. Now, entropy is a very difficult concept to talk about, and how we are going to define it and how we are going to discuss it is like a very small understanding of what it is. I've tried to speak to many engineers to figure this out, and they start getting into math. I'm like, no, you've lost me there. So what we are going to say is entropy is a measure of disorder and randomness. It is basically the law of the universe. Everything is going to get more and more chaotic, more and more disordered until the heat death of the universe. That's all tied up into entropy as well. It's the idea that if you were to take a stack of bricks and just toss them into the air, what is the likelihood that they make a perfectly formed wall? Basically 0. The more likely scenarios that they produce chaos or randomness. The wall is highly ordered and highly structured. That takes work and effort to put in. That takes energy. So energy is needed to basically fight entropy. In the end, though, there's a little caveat with this. Even though we're making more order in our cells and in our systems, we are still overall contributing to the greater entropy in the universe because we are putting off heat as a result of all of these reactions. And heat is one of those forms of energy that we have a hard time bringing back into the cycle and using again. So it's kind of like the dead end of energy. So even though we are fighting it to some extent, we are also contributing to it. It's a weird thing to think about. You don't have to understand that for the exam. So we use our energy from our food and from the sun to create all this order and fight that entropy. Any questions? Okay. We also talked about the different ways that we can get energy and our carbon sources, and we're going to hit on those again briefly today. We're going to talk more about them tomorrow in a little bit more detail. But the key thing is to know their prefixes. If you know that auto means self, so they are feeding themselves from carbon dioxide. Hetero means different. They're feeding themselves from different organisms. That's how they get their carbon for their biomass. For energy, we either get our energy from light or we get it from breaking down chemicals. Photo for light, chemo for chemicals. Now, where we get the electrons from also is a new category. Litho means we're getting it from inorganic molecules. Organo means we're getting it from organic molecules. So we are hetero, chemo, organotrophs. We get our energy or our carbon from eating other organisms and eating organic molecules. We get our energy from breaking down those same organic molecules, and we get our electrons from those same organic molecules. Plants are auto, photo, lithotrophs, basically, they get their carbon from carbon dioxide, their energy from light, and their inorganic molecules that they use to get their electrons is basically water, which is inorganic. So bringing all that together, we have our 2 groups that we're kind of going to hit on today. The phototrophy is more going to be next time. We have chemotrophy, where we're getting our energy and electrons from organic molecules or inorganic molecules through chemoorganotrophy or chemolithotrophy, and phototrophy is just getting it from light. You can be a photoautotroph, meaning you use light to fix inorganic carbon like CO2, or you can be a photoheterotroph. This is a weird one for us. Basically, they eat, but they get their energy from light rather than from the food they're eating. It's just kind of weird for us to wrap our minds around that. Like, why not just get it from the food you're eating? But life has a lot of different ways to survive. Any questions about those? This will make a lot more sense tomorrow. We're going to go through a couple of the different, like lithotrophs and photoheterotrophs, and we'll see how that really happens, how they get that energy. It's all basically in photosynthesis and the electron transport chain. All right, ready to keep going? There's some chemistry and physics here, and we got to talk about it, but we're going to talk about it in a nice way, not in a crazy way. So Gibbs free energy, you guys probably all heard that in general chem. And do you guys have to take physics here? Okay, you probably heard it in physics a little bit too. But basically, this is the energy available to do work after a reaction. And we talk about it in terms of, is the change in Gibbs free energy positive or negative? Because that will tell us, can the reaction happen spontaneously, just naturally? Does it have enough energy to go? Or does it require an input of energy first? So if delta G is negative, the process is spontaneous. This means the chemical reaction can go forward. It also releases heat. These are the exothermic reactions. And with this one, we're moving from a less stable to a more stable state anytime the delta G is negative. If it's positive, this is not going to occur spontaneously. This is an endothermic reaction, meaning it needs a big input of energy before it can start working. And as a result, we're going to go from a more stable to a less stable state. Because basically, we're taking something that doesn't really want to be broken apart, and we're forcing it into a situation where it's breaking apart, or we're adding things onto it that makes it less stable, and it doesn't want to do that. So we have to put in more work to get it to be in that state. This is when delta G is negative, it's basically saying this molecule is already not liking where it's at. It doesn't like something that's attached to it. It's not in a stable environment. It wants to come apart, or it wants to react as soon as possible. So it will require less energy to do so. Any questions about that? Like I said, super high level thinking here. We're not doing any math in this class. So what determines how a reaction proceeds, like the speed and the likelihood? It comes down to basically three things. Obviously, the intrinsic properties of the reaction. When we look at Gibbs free energy, it's a product of enthalpy and entropy. And so those we're not going to get into. We're supposed to, but that's chemistry. We'll talk about that not in the summer. Both of those contribute to the energy that we use to start a reaction. Also, the concentration of reactants and products. If I have more concentration of the stuff that's going to be reacting, there's going to be a greater likelihood of collisions, and collisions are what facilitate chemical reactions to occur. So the more stuff I pack into an area, the more likely they are to hit each other. Kind of like if you go into an elevator and it's packed full of people, you're going to brush into people. But if only one or two of you walks in, then that's going to slow down the reaction rate, because the odds of you hitting each other are a lot more unlikely. And then environmental factors. Obviously, temperature. We up the temperature, we move faster, more collisions. We lower the temperature, we move slower, less collisions. Pressure also does the same. It's effectively altering the concentration to very loose terms. And then things like salt and pH, those are going to affect how well our proteins work. Our enzymes are going to work, because all those charged particles from the salt or from the hydrogen ions or from the hydroxyl ions, those are going to affect the structure of the protein and potentially denature the protein. So those are the three things that are going to affect any chemical reaction in the cell. Any questions about that? All right. Now, we do something cool as cells. Life is super lazy. I'm sure we all understand that. If I can do something the hard way or the easy way, I'm going to do it the easy way. So that is exactly what cells do. They can look at a chemical reaction such as this here, adding a phosphate group to glucose. It requires a ton of energy input. This is a very high positive delta G. So this is going to take a lot of energy to do. But breaking down ATP and creating ADP and an inorganic phosphate group, that is the opposite. That's a very spontaneous reaction that releases a lot of energy. So the cell will look at that and say, why am I going to do them at two separate times and kind of waste this energy release and waste, my effort? I'm going to couple those reactions. As this reaction occurs, it's going to use its energy to take that phosphate group and add it to glucose. And then I can add those two reactions together and see that it's still going to be spontaneous. It's still a negative delta G, meaning this will still happen without an input of energy. And we end up with a net energy release still, usually in the form of heat, though. We can capture some of that. So it's basically saying, why am I just going to fight to do these and spend excess energy when I can couple it to another reaction and make it happen spontaneously? It's kind of like cheating. You're still paying the toll. You're still inputting energy, but you were cheating a little bit to get energy out of it in the end. It's kind of cool. So this is an example of what we call substrate level phosphorylation. This is basically saying we're taking an inorganic phosphate, basically a phosphate group that's on its own, and we're adding it to another molecule. Phosphate groups make molecules very unstable and reactive. So by adding it on, it makes that molecule more likely to have a reaction in the future. So if we want to do something to this glucose and we want to make it react readily, adding a phosphate group to it makes that glucose highly unstable, and it is increasing the likelihood that the next reaction it goes through is spontaneous rather than one that requires an input of energy. So kind of cool that we do that. Yes, exactly. That's the phosphotransferase system. That's exactly it. So this is all happening with an expenditure of a little bit of energy to move that phosphate group over. So yeah, the phosphotransferase system, when we input sugars, it does require a small amount of energy input, but when we see the result, we're actually releasing energy overall because the amount of energy released is greater than the amount of energy it took to make that change. Any questions about that? Okay. We are going to talk about 4 main energy carriers today: ATP, NADH, NADPH, and FADH, too. So these are what we call energy carrier molecules. They will gain or release small amounts of energy when we put them through reactions. And some of them are electron donors. These are what we call reducing agents, because if you remember, lose electrons, oxidize, gain electrons, reduced. So if they donate an electron, they're the reducing agent. If they are the one accepting the electron, they are the oxidizing agent. That's just redox reactions. You don't have to know how to do a redox problem. You just have to know the terminology. What is reduction? What is oxidation? And that I feel like we have a pretty good handle on. We all learn the various mnemonics to learn what's oxidizing and what's reducing. Which ones did you guys learn? Oil rig. All of you guys learned that. I was Leo Gur. So yeah, it's the old way of lose electrons, oxidize. Leo Gur, gain electrons, reduced. Yeah, maybe that's why we had the same chemistry teacher in high school. That's crazy. Yep. Miss Prater's the best. I love her so much. All right. So ATP is the main one we're going to talk about because it's kind of the main energy carrier of the cell, in the sense that it's the one that we've dubbed the main energy carrier. It's the one that we pay the most attention to. It's adenosine triphosphate. It's made up of an adenine base, a ribose sugar, and 3 phosphate groups connected to each other. I do this really cool lab with, or did, I'm not teaching high school anymore. I did this really fun lab with my high schoolers. I took a block and I wrote A on it, and then I took 3 smaller blocks and wrote P on them. And I took a rubber band, put it around A and 1 P, then another one around A and 2 Ps, and another one around the A and the 3 Ps, and I made them cut each rubber band in sequence and measure how far it flew across the room to kind of show the most energy is stored in between the bonds of the 2nd and 3rd phosphate group. Because effectively, we've got these highly, highly negative molecules here. And when we put a bunch of the same charge right next to each other, they want to repel each other. So we're kind of like charging up this battery and charging it up. And when we break that connection finally, it's like, oh, thank God, and it flies away and releases a bunch of energy. That's why we can capture so much from it from ATP. So that's just kind of a fun way to think about it. And there is more energy in the other bonds too, but it's just significantly less than between the 2nd and the 3rd bond. We will often use those other bonds for other energetics reactions. It's just not something that we need to talk about here. Something cool though, magnesium is having its day in the sun in the health world right now. And one thing that I didn't realize until very recently was magnesium helps stabilize our ATP molecules and ADP to some extent, but it's most important for ATP because in this adenosine triphosphate state, it really does not want to stay in that state. It will dissociate very readily. So we add in a magnesium to cause all the phosphate groups to kind of turn on themselves and connect back to the adenine, and it stabilizes the whole structure. So it allows the ATP to be stable and remain in the cell until we need to do the reaction with it. Just kind of cool to think about. Any questions about ATP? I feel like you've heard ATP a billion times at this point. All right. Now, ATP is only a medium-sized energy carrier. We think about it as the top, but technically, glucose is one of the tops. It has a lot of energy stored in its bonds. That's why a lot of organisms use it for food. It's the preferred food source for cells because it has the highest energy potential there. And then we have other types of sugars, and ATP is right here in the middle. And then we kind of get less and less as we keep going. So there are other energy carriers that are better than ATP. We've just evolved to make ATP one of the primary ones that we use. In the end, it all comes down to evolution, and whatever was good enough at the time, the cell learned to live with, and we built around that. Evolution is not about making perfect, it's about making good enough for right now. If I can live, I will serve. I'm winning the battle of life here. So ATP transfers energy in 3 main ways. It can release that inorganic phosphate group. It can release 2 inorganic phosphate groups, or it can be used for that substrate level phosphorylation, and it can just transfer one of its phosphate groups directly onto another molecule. And this happens with all of our other trinucleotides, ATP, GTP, CTP, all that stuff. We can use them all for energy, and we do, depending on the circumstance. ATP and GTP seem to be the ones we use most often, though. Any questions about that? Okay. I'll wait 1 second. I'm seeing a lot of people writing still. I think we got time today to take moments. Alrighty. Let's go. This surprised me when I learned it the first time. NADH carries 2 to 3 times the energy of an ATP. Often when we talk about this whole metabolism and central metabolism, we're like, oh, the goal is ATP. That's the most energy-rich molecule. And we kind of ignore all these NADHs, but they're actually where it's at. And in the end, we kind of see that. The whole first two steps, glycolysis and the TCA cycle, are all about making a bunch of these energy carrier molecules. And they move into the electron transport chain. And that's where we get the huge payoff of ATP. We can make 32, 34 ATP from all of the NADHs that move over into the electron transport chain. So they are very energy rich molecules. And they are also technically a nucleotide. So same basic chemical structure as DNA and RNA and ATP. They donate and accept electrons. So NADH is the reduced form and NAD plus is the oxidized form. So NAD plus is the one that's lost an electron. NADH is the one that's gained an electron. And overall, it's going to consume 2 hydrogen atoms to convert between NAD plus and NADH. And one thing I want us to kind of consider is we're going to be talking a lot about hydrogen ions, and you're thinking, oh, but I thought the point of this was moving electrons. We're also going to talk about those a lot. If I have a hydrogen ion, this is basically just a proton. If I add 1 electron to it, then this makes a whole atom, and this will make a complete hydrogen. So if we're talking about moving or passing around hydrogen ions, we are also moving around the electron that pairs up with it. That's the idea behind it. I'm sorry if this seems simplistic, but I feel like sometimes we don't make these tiny little brain connections, and sometimes those kinds of connections can give you aha moments for harder things down the road. That's why I try to focus on those little things too. Okay, are we ready to keep going? There's also FAD, flavin adenine dinucleotide. Once again, it's a nucleotide, a nucleic acid. And it's another coenzyme. It transfers electrons. The reduced form is FADH2. The oxidized form is FAD. And it takes 2 electrons and 2 protons again. Now, this does not carry quite as much energy as an NADH because it's a weaker electron donor. So it's harder to get the electrons from FAD than it is from NADH. Or I'm sorry, from FADH2. So if we're going to be using FADH2, we have to combine it with something that really, really, really wants to steal those electrons, because it gives it up so weakly. It's kind of like, no, I kind of want to hold on to these. You have to pair it up with something that says, no, I want those electrons. I will get them one way or another, like oxygen. Oxygen is probably the strongest electron acceptor. It's very electronegative. It wants those, and it doesn't feel complete without them. So it will take them at all costs. And that's why it's so dangerous in our cells too. Any questions about that? All right. Now, just a word about enzymes again, because everything we're going to talk about today is not possible without enzymes. And literally, even the most basic reactions required for life, if we waited for them to occur on their own, it could take thousands of years for that reaction to happen on its own. Enzymes make them happen hundreds of times a second in all of our cells. So they're very necessary, and they work by lowering the activation energy. Every single reaction does need some input of energy to get started. Some reactions just give more energy afterward than what you put in, so they're exothermic overall. But every single reaction has kind of a hurdle, a toll you have to pay before it gets going. And what enzymes do is they lower the fee. They make you pay a little bit less than that toll. They lower the size of the hill, which makes it easier to climb over, basically. And they will couple energy yielding reactions with energy requiring reactions. And a lot of times what enzymes do is they are proteins, so it's all about the shape. If they are in their correct shape, then the reactions, the reactants that they're going to be working on are going to fit in just the right way that the enzyme has them basically have a collision in just the right spot for the reaction to occur. because it's not just that two chemicals have to collide. They have to collide in the exact right orientation for that bond to happen, and with the effective force, the energy required to make that reaction happen. So it's a lot of little things that have to come together for every reaction to occur. And enzymes basically say, I'm going to take away some of those hurdles and I'm going to put you in the right position and make it happen for you. So they also have allosteric sites. You guys remember that term? I think it's fun. I don't know. We talk about enzymes, and it's more fun to connect them back to health. And when we have competitive or non-competitive inhibitors that really affect the health of the organism because of what enzyme they're affecting. Allosteric sites are sites that are not the active site. So something else can bind and affect how that enzyme works, either making it work better or worse. One really interesting example is cyanide. It's a competitive inhibitor of some of the proteins in the electron transport chain. So that kind of shows you how important the electron transport chain is to keeping us alive, that you could take a cyanide pill and be dead in seconds because it's inhibiting the electron transport chain enzymes. So just something to think about. Bringing it back to the bigger picture. All right. We're going to get into it now. So we're going to talk about 3 forms of glycolysis, because bacteria can do other forms of glycolysis. And we're going to talk about the TCA cycle. Next class is electron transport chain and photosynthesis, because they're all, those two are effectively the same thing, just flipped. All right. Now, bacteria can break down polysaccharides, they can break down disaccharides, they can use energy from monosaccharides, but basically, a lot of the goal is make pyruvate. So whatever's coming in, either we build it up to make a pyruvate or we break it down to make a pyruvate. And from there, I'm going to put it through the TCA cycle and the electron transport chain and get my energy from it. So while we think glucose is the main player here, it's really pyruvate. That's what the cell really wants. And we just make pyruvate the easiest from glucose because we just break it in half and there we go, 2 pyruvates. We can also eat lipids. We can break down proteins and use those for energy. There's lots of different compounds that we can get energy from. And sometimes when we break down these lipids and amino acids for energy, the intermediates that are created, we can then funnel those away for something called biosynthesis. And we can build up other molecules that we need in the cell. And that's Monday. We're going to talk about that Monday. It's a fun, short one. They can also break down aromatic compounds, so compounds that have ring structures. Those are very hard for our cells to break down, but bacteria can. So oftentimes, that's why our ruminants, like cows and goats and all that, can eat grass and we can't, because they have more of the bacteria that can break this stuff down. And it'll help them digest it for them. And that's why they have that extra stomach pouch that contains all that bacteria to help them break it down. It's fun stuff. Any questions about that before we dive in? This is basically just an overview of like, hey, this is where we're heading. All right. There's 3 main catabolic pathways that we're going to talk about. Fermentation, the delicious one, respiration, and then photoheterotrophy. This is basically breaking down food using light energy rather than other types of energy. So we are mainly going to focus on these 2 today. This one we'll talk a bit about next class. All right. And we're going to make it easy. There's a lot of reactions for fermentation. I'm going to show you a scary picture. You don't have to know the scary picture. It's just there for you to help you understand that it's complicated. And that's all we have to know today is it's complicated. All right. One of the things we do have to talk about, though, is these 3 pathways of glycolysis. All of them are going to follow a similar pattern. There's going to be an activation step, an energizing step. We got to pay the energy toll before we can get the benefit from it. This is often that step where we add the phosphate group to the glucose or to the sugar. That is energizing enough. We've activated it and it's ready to go now. Then we're going to move into a phase where we're going to be slowly extracting the energy from these molecules. And then we're going to get the yield phase where we kind of like get the payout. I think I mentioned this later, but I feel like it fits a bit better here. There is a reaction where we can immediately take glucose and break it down into the end products in one step. We don't do that for a lot of different reasons. One is, if we do that, the energetics of the reaction, the physics of it all, we will end up releasing way too much energy as heat. Because if we look at our whole reaction in general, we're taking glucose, we're adding oxygen to it, and we're making carbon dioxide and water. What type of chemical reaction is that? What category of chemical reaction? This is the key here. When we add oxygen to anything, what are we doing? We're burning it. This is a combustion reaction. So literally, if we tried to do a whole breakdown in one go, we might produce way too much heat and the cell could like catch on fire. So we don't want that. We want to be careful with our fire. We're going to play with it a little bit here, and we're going to play with it in a smart way. So rather than doing one step, we break it down into smaller steps. This minimizes how much heat is released and basically lost, and allows us to pull out more energy from all of those bonds. Another reason why is, this gets back to more anatomy and physiology, but it applies here for the cell as well. When you have reactions, every single reaction in the world is reversible. Literally every single reaction. It's just how much energy you got to put into it. In the cell especially, as we are breaking down these products, if I only have two steps, the likelihood of it reversing back and us wasting all that energy that we just put in is pretty high. We don't want that to happen. So if I do one intermediate step and then immediately take that middle thing that I have no use for aside from continuing the reaction, and I do another reaction on that intermediate, I've now removed that intermediate from the equation. It can't go backwards anymore. So it ensures that when we're making energy, we're constantly flowing towards the end goal, and we're not getting caught back up going backwards and wasting our time and our effort. I'll have to find a picture and post it, but there's some really good pictures of this using kind of like a water and a turbine example. I think some of your 106, 107 books use that, but basically we want the reaction to keep going, and by removing the intermediates to keep it going, we prevent it from going backwards and from causing us problems. So that's why, two reasons why we break all of this down into so many little steps, mainly because it gives us more energy. All right, so let's jump into glycolysis. We're going to talk about glycolysis in a different way than you've heard before. Hopefully not in as complex of a way as you've heard before, because I'm not going to make you memorize all the little chemicals in between. The first one that we're talking about is what we know as glycolysis, but is also called the Embden-Meyerhoff-Parnas, or EMP pathway. This is traditional glycolysis, the one that our cells do, that plant cells do, all that. Bacteria can also do these other two pathways, the Entner-Dauderoff, or ED pathway, and the pentose phosphate pathway, which is also known as the pentose phosphate shunt. So it's all kind of building off of this main glycolysis here. And I'm going to bring it up here. Yeah. Yes, they do all three. Yep. Basically, think about it as EMP is done by everybody, and bacteria are special, and they can do other ones as well, depending on what they need and what their environmental state is. Because if we look at their three yields, the normal glycolysis pathway is giving us 2 ATP and 2 NADH. Both of those are energy molecules. We make them primarily to get energy from them. Then in the middle one, the ED pathway, we have ATP, NADH, and one NADPH. NADPH can be used for energy, but more often what we use it for in the cell is biosynthesis, what we're going to talk about on Monday. Basically, we use that to make more molecules, like more proteins, more DNA, things like that. So if we're just in a I'm starving, I need energy mode, we probably want to do the first pathway. If we're like, we kind of need energy, but we also kind of need to build stuff right now, we'll do the ED pathway. If you look at the PPP, it's 1 ATP and 2 NADPH. So this is a pathway where the cell says, I'm flush with glucose right now. I got way more energy than I need right now. So I don't need to waste my time making NADH because I don't need it. What I need is NADPH to make a lot of molecules to do anabolism. So it will pick and choose which one of these to use based on what the needs of the cell are. Energy or stuff to do anabolism. That's the idea behind it. That's the biggest picture that I want you to take from this, more so than the nitty-gritty details. You'll get nitty-gritty details in microbial phys. He'll want you to memorize a lot of this. It was not fun. All right. But I got a really good understanding of glycolysis. All right, let's jump into it. So we'll talk about the EMP pathway first, the most common one that everything does. And this is going to occur in the cytoplasm. It can happen whether or not there's oxygen around, and it's really 10 distinct chemical reactions, 10 steps. But we're going to break it down today into 2 groups of steps and talk about those from a higher perspective. And in the end, we're making NADH and ATP. You've heard this before, right? Now, really something kind of interesting to think about that might blow your mind is our cells do this in our cytoplasm, but chloroplasts and mitochondria are also cells that need their own energy. And so they are doing this in their cytoplasm equivalent to make energy for themselves as well. So we've got 2 cells doing this in your own cell at the same time. Very crazy. So let's jump into it here. Stage 1. This is the glucose activation stage. We got to get the glucose ready to be broken down because it's not in a good state in just glucose form. Usually, this is where we add the phosphate group to it. So usually this phase happens as the sugar is coming into the cell because that whole PTF system, as the sugar comes in through its transporter, it gets the phosphate group added before it even enters the cell. And this turns the glucose into fructose 1,6-biphosphate. I'm not going to ask you to give me that name, so don't stress about that. We don't have time to spend wasting on that. But just know that this new molecule that it's converted into gets broken down into 2 different molecules here. Technically not the same molecule. We make 2 different ones. So glyceraldehyde 3-phosphate can just move on to the next part. But dihydroxyacetone phosphate has to be converted into glyceraldehyde 3-phosphate before it can move on. And if you're curious, if you just want to understand what's going on, the book has some good pictures that break down the steps. You don't have to know them, but reading through it sometimes just helps you get a better understanding of what's going on, and it's interesting. Maybe only to nerds like me, though. So far, we've done no redox reactions. So we're not collecting energy yet. In fact, this part, we're paying energy still. This is the part where we're paying those 2 ATP up front. All right, any questions before we move on? And then the second half of it is the energy yielding stage. The glyceraldehyde 3-phosphate goes through a series of reactions where we produce 2 NADH molecules and 4 ATP that we make through substrate level phosphorylation. So just passing phosphates around. So remember, it's only the G3P, glyceraldehyde 3-phosphate, that can go through this part. So the other one, the other product, has to be converted to G3P first. Part of the energy requiring phase of the whole thing. And this is about as deep as I want you to go on the EMP pathway. All right. I'll wait another moment, seeing people writing a lot. Okay. This is the whole thing. So stage 1 requires the energy. Then we take our 2 products and put them into the energy yielding phase. And that's just a nice summary picture of it all. So let's move into the ED, the Entner-Doudoroff pathway. Doudoroff, I don't know. ED pathway. So this is probably an older pathway than the EMP. Even though the EMP is used by most cells, this is probably the one that had the foundation, and then it was modified to become the EMP, and all cells took that model later. So there's fewer steps of substrate level phosphorylation, and as a result, we make less ATP, fewer ATP. And this is basically only seen in prokaryotes. We still have to activate our glucose, and then we cleave it into pyruvate and glyceraldehyde 3-phosphate. So one of them can go through the normal EMP pathway, and we see here that it does. So here's the summary of them. We just saw the EMP pathway. With the ED, one of the molecules that we make can be funneled back into the EMP pathway and go through the energy yielding phase. That G3P can. The pyruvate can directly enter the TCA cycle, which we're going to talk about in a moment. TCA cycle is also the citric acid cycle, the Krebs cycle. There's a lot of names for it, and everyone uses a different name based on what path of biology you use. Microbiologists use TCA for tricarboxylic acid cycle. Now, this pathway makes 1 ATP, so total it's 1 less than the other pathway. 1 NADH, so that's less than the other pathway, but it makes 1 NADPH that we didn't make before. Cool. Not too bad, right? So this is the goal. I'm trying to make it like accessible. All right, so this is just that summary picture a little bit better if you need it, or bigger if you need it that way. All right, and lastly, the pentose phosphate shunt, or the pentose phosphate pathway. So this is similar to the ED pathway. In fact, it doesn't have a starting point on its own. Its starting point comes from an intermediate from the ED pathway. That's why it's a shunt. It's basically, a shunt is something that bypasses a primary route. So instead of going down its own, it's its own side passageway for the ED pathway. It's going to take 6-phosphogluconate, and it's converted into ribulose 5-phosphate. This is going to produce a bunch of sugars on its own. You don't need to really know that step. You just need to know that it is a shunt from the ED pathway, and it produces sugars that can go into biosynthesis, and this is mainly used for biosynthesis, not energy production. And so we make 1 ATP and 2 NADPHs. That NADPH is what's needed for biosynthesis as well as those sugars. NADH is not made here. You do not have to know all that stuff there. Don't do it. Save that for a normal semester when you have time. Any questions about the 3 different forms of glycolysis? All righty. So fermentation is kind of like an intermediate step. It's saying we can't quite go into the TCA cycle yet, or we need to do other things that require fermentation, but they all come basically after glycolysis. And it's completion of catabolism without the electron transport chain. Usually this is because we don't have that terminal electron acceptor. We don't have oxygen, we don't have nitrogen, we don't have sulfur, something to take that electron at the end. So what happens is the cell will start to accumulate a ton of NADH, because the point of the NADH is to give electrons to the electron transport chain. If we have no electron transport chain, we're going to start to accumulate a ton of this in the cell, and that's kind of a waste. And even more so because now, if I have too much of this, my glycolysis reactions won't happen, and I won't make any ATP anymore. And I'd rather make a little than none. So we use fermentation to restore NADH back into NAD+, so it can go and we can do glycolysis again. So technically, fermentation does not generate any energy on its own. It doesn't make any ATP, but it lets the cell continue to do glycolysis to keep making ATP. So it's a helpful step. Now, a lot of organisms can survive using fermentation. Mainly are bacteria that can do anaerobic or aerobic respiration, or our anaerobes do this often. A lot of organisms in our gut do it. And it's just another way of surviving, basically. Now, there's 4 different overall types of fermentations, and you do have to know these. So homolactic fermentation, break down the word to remember what it is. Homo means the same. Lactic means we're making lactic acid. So I'm making 2 things pretty much every single time I do a fermentation reaction. In this case, both of those things are lactic acids. In ethanolic fermentation, I'm making 2 things again. One of them is ethanol and 2 CO2s. Now, this is the one that we are probably most familiar with if you've ever done sourdough, if you've ever fermented beer or anything like that. That's ethanolic fermentation. It makes all the stuff that makes you drunk, or delicious bread that makes us happy. A lot of fermentation is just a very happy thing. We get a lot of good stuff from it. Then there's heterolactic fermentation. So hetero is different. So we are making one thing is lactic acid, but what's the other thing? It's going to be different. In this case, it's lactic acid, ethanol, and CO2, because ethanol and CO2 always kind of go together. And then we have mixed acid fermentation. Which of these acids are produced, how many of them, how much of each, it depends. But we're going to make some combination of acetate, formate, lactic acid, and succinic acid. And then we're also going to make ethanol, hydrogen gas, and CO2. This hydrogen gas makes it super stinky. Any questions? We're going to talk about this in lab too. Mm-hmm. Yep. Yeah, you don't have to say 2 of this and 2 molecules of that. You can just say overall it makes lactic acid, or it makes ethanol and CO2. Let's make it easier on ourselves right now. All righty. Now, a lot of these, lactic acid, all of these from mixed acid fermentation, they are our organic acids. They are our membrane permeant weak acids. So if we limit where the bacteria's environment is, they're in a container or they're in a jar or whatever it might be, over time, these products are going to build up and build up and build up and sneak into the cell, because they can pass into the cell without a problem. And it's going to make the cells more and more acidic. They'll have an acid response that occurs, but eventually they're going to be overcome by the acid because they have nowhere to go, and their environment just keeps getting filled with more and more acid as they're trying to do fermentation. So eventually, the products that they create end up killing the bacteria. And we humans learned this, and we used it as a way to preserve food. So if I'm killing the bacteria aren't going to be able to make my food spoil. So I let the bacteria ferment it, kill themselves, and now I have a shelf-stable product without refrigeration needed. So just kind of a cool thing that humans have done. So you don't have to understand this, but you can see all the different types of pathways to get these equations and all the cool stuff that we make as a result. It's not just delicious things like bread and alcohol. It's like kimchi and sauerkrauts, cultured butter, yogurts, and also we can get a lot of industrial products too. So fermentation is something that is done in industry all the time to make things that we need for either food production or for medicine or for other industry or for research. So it's a very important thing, and we've learned how to kind of tame the bacteria and get what we want out of them. All right, let's talk about after glycolysis here. So after we've done that whole step, if we have a terminal electron acceptor available, like oxygen, like nitrogen, like sulfur, something like that, we can move into the TCA cycle and the electron transport chain. So the first thing we have to do is take pyruvate, which is the end point of all of our glycolysis pathways, basically, and we have to convert it to acetyl-CoA. And this is done by an enzyme called pyruvate dehydrogenase complex. So we take the pyruvate and NAD plus and CoA, and it becomes acetyl-CoA with some release of CO2 because we're cleaving off one of our carbons. And we also steal some energy from this whole process because we cleaved off a carbon. And so we get an NADH and a hydrogen ion out of it. We're basically transferring around some electrons, and one of them got passed on to NAD, and it's going to get used later on. You guys have probably had to memorize the steps of this whole reaction already, haven't you? All right. A lot of you are like, cool. So it's the acetyl-CoA, not the pyruvate, that technically enters the TCA cycle. So it's a pre-step that has to happen before we can get into that whole Krebs cycle process. In prokaryotes, this is going to occur in their cytoplasm. In eukaryotes, this is one of the first steps that happens in our mitochondria. And so this is going to occur in the matrix of the mitochondria, which is basically the term that means cytoplasm of the mitochondria. I think it's the stroma for the chloroplast, if I remember correctly. Now, acetyl-CoA is also required for a lot of fermentations. So the cell will convert pyruvate to acetyl-CoA and then be like, oh, I don't have any oxygen. Okay, well, I'll take that and I'll ferment it. So there's a lot of ways that we can do fermentations. Now, we're going to mainly discuss today oxygen as the terminal electron acceptor, but next class is when we're going to talk about the other forms through anaerobic respiration. Because we're using oxygen here, this is all part of aerobic respiration. Ready? So, do you have to memorize all this? No. But just know that a lot of these are going to come up again on Monday. So you might want to be familiar with the key molecules here that are the key intermediates. You don't have to know which comes in which order, but be familiar with the names. So we have our first step where pyruvate is converted to acetyl-CoA. We take our 3-carbon molecule and convert it to a 2-carbon molecule, and that releases a CO2. And then acetyl-CoA can bond to oxaloacetate. I always have a hard time with that one. And it becomes citrate. That's why it's the citric acid cycle. And then citrate is going to slowly get broken down. We're going to take all those carbons and slowly cleave them off and release them as CO2. And every time we do that, every time we break one of those bonds, we're releasing a little bit of energy and we're harnessing it in the form of NADH. We're capturing the electrons and saying, these are good, we're going to use them for later. Because anytime we have electrons moving through something, that's literally electricity. So we're storing up the electrons to take them to the machine that can use the electricity. And eventually we also make some ATP, and some FADH too. So it's not just NADH that we're making here as we're collecting all of these electrons. Now, what I want you to get from this one is that lots of things can enter and be converted into acetyl-CoA. We can take sugars, lipids, peptides, that lignin, kind of like woody material that bacteria can break down, and we can all get them to be in a form that can be converted to acetyl-CoA and then enter the TCA cycle. So a lot of times we think that it always starts with glycolysis for metabolism, but not always. A lot of times when we're eating other types of food like fats or proteins, they don't enter at the beginning of glycolysis. They're going to enter here somewhere in the TCA cycle, either at the beginning. Sometimes they can even be converted and enter in the middle of the pathway, and we can use those intermediates to make more proteins and things like that. And the one thing I also want you to note is that the enzyme names match what you're creating. So we have citrate here, we make it right here, and what enzyme makes that? Citrate synthase. Then over here we make 2-oxoglutarate, and we have, this one's a little bit not necessarily the name, but it's isocitrate dehydrogenase. We take isocitrate and we remove a hydrogen from it, and that makes alpha ketoglutarate there. So just know that if you do have to memorize it, there are cues that make it a lot easier. All right. So moving on, it's going to take that, or we already went through this. Why am I saying that again? We're basically, I pointed this out already. I just wrote it out. Sorry. This is where I wanted to get to. What do we make? Now, remember that only a pyruvate, an acetyl-CoA, can come into the cycle. So we're talking about per pyruvate, but when we start with glycolysis, we're talking about a glucose. So often when we tally up final numbers, we're saying we're doing this whole process twice. One for the first pyruvate from glucose, the second time from the second pyruvate from glucose. But we're going to make 3 CO2s because pyruvate is a 3-carbon molecule, and we are wanting to break apart the bonds between those and release those carbons. We don't want them anymore necessarily. We want the energy in them now. We're going to make 4 NADH and 1 FADH2, and that's all through redox reactions. And then we make just 1 lowly ATP. So this is not a big ATP producer. The whole goal of this is harvest the electrons so we can put them to the electron transport chain. Sometimes the cell will make GTP instead, but we just use ATP to make it easier to understand. So overall, per glucose, it's 6, it's 8, it's 2, it's 2. Makes sense? This has been a good review day, right? Like, not too many new things, right? Just the 2 new pathways, hopefully. The rest of this, I know you've heard it in high school. I know, that was a long time ago. All right. Now, after we do the TCA cycle, we take all those electrons and we have to continue the process of using the energy stored in them. Basically, they're in a high energy state, and we're going to let them fall to lower energy levels and collect the energy they release as they fall. And that's in a process called oxidative phosphorylation because we tie it to making ATP. And in the end, all of our carbon is released as waste to us. But if you're an autotroph, that's carbon that you want. That's carbon you can use for energy and biomass. So when you think about it, all of the weight on your body, if you try to lose weight, where does the lost weight go? You're breathing it out. So you're just taking the weight that's on your body and breathing it away. And plants then, how do they go from a seed to a gigantic plant? All of their mass is just taking that CO2 from your breath and converting it into a physical form again, which is just cool to think about. All right. This is what we're going to hit tomorrow. And we're going to get all into the proteins. We're going to talk about ATP synthase and how that works. And we're going to talk about different forms of the electron transport chain for different types of organisms. Last thing I want to mention is on Monday, we're going to look at this TCA cycle again and see how we can take these intermediates and turn them into amino acids and form proteins from them or lipids from them, and use this whole pathway as both a catabolic pathway, getting energy from it from breaking it down, and an anabolic pathway where we can also use it to create molecules in the cell. Any questions about that? All right. Let me pause this and then just a couple reminders.